Natali is a biologist in the branch of science support with the U.S. Fish and Wildlife Service based out of Falls Church, Virginia, where she provides scientific expertise to inform endangered species listing decisions, recovery actions, and species status assessments. Previously, Natali was the National Recovery Lead for 10 federally listed species. Prior to joining the service, she worked as a biologist for environmental consulting companies, research institutions, nonprofit and federal conservation agencies in 3 countries, Peru, United Kingdom, and United States. Natali is an AAUW American Dissertation Fellow and National Socio-Environmental Science Synthesis Center Graduate Fellow. She led an interdisciplinary team to investigate how public engagement with biodiversity influences policy and how it relates to conservation success. Please join me in welcoming Dr. Natali Ramirez-Bullon talking to us today about refined approaches to improving efforts to conserve plant biodiversity. Go ahead, Natali.
Thank you, Elizabeth, for the introduction, and I'm very happy to be here. This is my dissertation talk, so I hope you don't find it too boring. So welcome. I'm going to start with a sad part. So we know that by 2050, the world's population is projected to approach 9 billion. With more people, there is the need to develop more land, and that results in conversion of natural areas for agriculture expansion, followed by overexploitation of species. Most mega biodiverse countries have low to upper middle incomes, resulting in a shortage of funding and expertise to implement conservation actions. Creating conservation guidelines can help developing countries comply with international conventions and in some cases result in poverty reduction by ensuring the protection of natural resources. Distinguishing species that require immediate conservation actions to prevent extinction from species that are rare but demographically stable can help make conservation decisions when resources are scarce, as is in the case of many countries with high biodiversity. These challenges inspire me to develop my dissertation goals. I wanted to develop a quantitative framework that will allow scarce conservation efforts to focus on species that needed it the most, a sort of triage, because we only have a good understanding of a few species. For example, the IUCN Red List, an important reference for the threat of a species, is still heavily biased towards vertebrates, with plants only being assessed by 9%. Therefore, a method that could help us distinguish plants with greater conservation needs will facilitate conservation and policy decisions. For the US, about 57% of listed species under the Endangered Species Act are plants, but they don't receive a proportional amount of funding. If we look at the graph on the left, we can see on the y-axis, this is the number of listed species per taxonomic group, and about 57% of those are plants. And if you see on this graph, this is the investment per species for the years 2009 to 2011, and plants only receive about 4% of all conservation funding. How to assess when plants species are recovering or declining to extinction? To me, the best way is to assess their population growth rates. Demographic information is not available for most rare species, and it takes a lot of expertise and many years to collect. Demographic studies require to mark and follow individuals for a few years in order to have enough data to calculate their vital rates. And it also requires a very good understanding of their life cycle. Most studies also are done in common species, so we don't have a lot of information for the rare species that we need to assess, for example, for listing. And when we think about rare species, I don't know, if you put on Google rare species, most of them are going to be very charismatic, like the pictures I put on this slide, but not all rare species are endangered. Rabinovich proposed this framework, 7 forms of rarity, to understand causes and consequences of being rare. She emphasized local population size or abundance, like the one that this access, and habitat specificity, either narrow or wide. Species can become rare for different reasons. So having this framework in mind can help us identify what are the causes and consequences of their rarity. For my dissertation, I focus on rare species in the community ecology sense. That means like the species that are low in relative abundance, which is not the same as endangered or threatened. Because low abundance is the precursor of extinction, but not all low abundance species will decline to extinction. Some low abundance species might persist stably. If plants persist stably at low abundance, one of the mechanisms that will allow it is a strong negative density dependence. Population growth rates are greater when a population size is small. As you can see on this graph, if for some reason the population size is going to be decreasing, the population growth rate of the species is going to increase. So that will make them more resilient and more likely to overcome that impact that reduce their population size. And there have been mathematical models and observational studies that propose this mechanism too. For example, Jenny et al. Proposed that a strong negative density dependence promotes the persistence of rare species using mathematical models. Another study followed that hypothesis using observational data, but there was not been a field experiment that tested for negative density dependence. And that's what I was planning to do. For example, Fowler et al. Found a demographic signal of negative density dependence in a common species. She removed conspecifics, so here are adults removed, and also added more conspecifics on their plots. And she found evidence for negative density dependence. When conspecifics individuals are added and population growth rate increases, somebody had their microphone on. Well, anyway, so you can see how population growth rate increases when adults get removed and decreases when more conspecifics are added. With that, I predict that low abundant species will have a greater signal for negative density dependence than common species, as you see in this graph. So rare species are going to have a very clear signal of density dependence compared to common species. Another mechanism that could drive negative density dependence is herbivory, because of decreased predation rates when a species is low abundant. So species that are low abundant in landscape are going to experience less herbivory because there is more probability that insects will specialize in the consumption of common species like these ones. Considering these mechanisms that would allow a species to remain stable at low abundance, we could use traits associated to greater competitive ability or herbivory to distinguish low abundant species that are at low risk of extinction, the ones that are going to remain over time at those low abundance. There have been previous studies that look at traits. For example, Cadot defined rare species as those identified as threatened and endangered and couldn't find a trait that could distinguish them. And Espeland also used the Rabinowitz framework, but she only used reproductive ecology traits and she excluded common species. So she couldn't make that good conclusion without that information. It has been hypothesized that a species that experience a stronger negative density dependence will have traits related to greater competitive ability. And this is because they are very good competitors within each other. So interspecific competition is greater than interspecific competition. These traits are rapid growth, large seed mass, greater maximum height, and reduced investment in reproduction because they have to allocate more energy to rapid growth. I will present now my research objectives. First, I wanted to test the hypothesis that rare persistent species have a stronger density dependence than common species and identify traits associated with a strong density dependence because those might indicate demographic stability. So for my first objective, I used long-term plant community surveys to identify 3 species pairs of common and low abundant species native to the longleaf savanna. I selected species that persist at low abundance for many years, at least 8 years. The respective co-occurring common species. I conducted density manipulations in the field for co-occurring species. So I had 3 general 6 species. I set up seed and seedling plots. And I'm going to show you next my example of how I did it. For 4 species, I grew enough plants in the greenhouse to have 4 density levels. For the other species, Eupatorium, the eupatorium part, I only had enough plants to do 3 levels of density, low, medium, and high. For all the species pairs, I more than double the density observed in the field. For my experiments, I had the following expectations. I was expecting to see a greater slope, low and high density dependence, and that will indicate how strong negative density dependence is happening. And also, I expected to observe more herbivory in common species compared to low abundant species. See faster damage on like the common species in general, especially at high abundance, high density. Overall, I also look at survival. Overall survival to harvest the individuals was really high for all the species. Eupatorium was 92%, Solidago had 95% survival, and Symphyiotrichum species pair had an 86%. I did not find any difference between the species abundance or the density treatments. So there weren't difference in survival, and it was really high. I found no significant effect of density on the average biomass or abundance category, and that was kind of sad, but whatever. And I didn't find interaction between density and abundance category for any species pairs, which was surprising because I more than doubled the density of the individuals. As you can see in my plot, like they are growing on top of each other, but that still didn't affect their average biomass. So here, the black plots represent the low abundant species and the gray ones, the common species, and these are my 3 species pairs. I did not observe any significant difference for them, so no signal of negative density dependence. I was expecting also to observe greater allocation to reproduction or greater average number of flowers in common species compared to low abundant species. I found that the 2 species pairs, the low abundant species didn't produce any flowers. As you can see, there is no black box next to it, only gray. For the Symphyiotrichum pair, I found greater average number of flowers in common species compared to low abundance, but there wasn't a significant difference between density treatments. When examining herbivory damage, I did find greater herbivory on common species compared to the low abundant species at all densities. This agrees with some of my predictions because I can rule out the negative density dependence is stronger in low abundant species. I look at traits associated with a stronger competitive ability in these three pair of species. I focus on comparing maximum plant height and specific leaf area because they are positively correlated to the fast maximum growth, which will make them stronger competitors. I expected to have or to observe a greater value of plant height and species SLA in low abundant species and uncommon species because that will give them an edge than common species. I found that the low abundant species that persist over time were significantly taller than their closely related common species. For each species pair now, I will present the results of a specific leaf area. So for here, I was a little right. I found that the specific leaf area was significantly different for Eupatorium and Solidago, but in different directions. I was expecting that the low abundant species will have a greater specific leaf area or SLA than the common species because they will grow faster. But I did not find that. For Symphyiotrichum, I didn't find that anyway, or the difference wasn't significant. So my takeaway of this first part, which was a very tedious experiment, I found no significant difference in juvenile survival and no evidence of a stronger intraspecific competition in low abundant species compared to closely related common species. There was a slight evidence that low abundant species experience less herbivory damage than the closely related common congeners. There is a small evidence of the potential of traits associated with low abundance species that persist stably. So this second finding motivated my second question. And first, before I transition to the other one, I wanted to point out, this is like a summary table. The cells that are highlighted are the ones that match my predictions. But as you can see, it's a mismatch of evidence that supports what I was predicting and the other part that didn't support for my 3 pair of species. Okay, but this is limited, right? Because I only look at 6 species, 3 pairs. Another way is to look at all species that have data available, which is what I did for my second chapter. What species level trait values can distinguish low abundance species that are declining from the ones that are stable. For that, I needed to have demographic data, abundance data, trait data, and luckily there are some databases that have that information. One of them is the Compadre database and curates matrix demographic models of plants. And in 2021, they had 758 species worldwide. Another database is the TRI database, which curates data for 148,000 taxa and their respective traits, such as maximum plant height, seed mass, specific leaf area, and more. Because I thought the demographic data was the most limited because it takes expertise and time, many years to collect, I started by including all the species where we had demographic data, except those that are invasive or grown in a greenhouse. For that, I realized that demographic papers don't usually include any information about the relative abundance data, so that was a journey. I read every paper on demography for that in order to classify them as either low abundance or common. To compare trait values on those species. So I wanted to get their demographic data, know which ones had increasing population growth rates, stable and declining. And then I went back to look at the trait data to see if I could find some. I only found enough information to compare 5 traits total, which was surprising. There was a very low overlap of species with that had demographic data and trait data. So I had to examine each trait separately. You can see by my number of species that I have data for. I couldn't evaluate them at the same time either, but I did test for phylogenetic signal, and none of those had phylogenetic signal. So all the analysis that I'm going to show don't consider phylogenetic or don't control for phylogenetic signal. I found that for low abundance, and we're going to start with plant height. I found that for low abundance species, those with decreasing population growth rates were 73% taller than species with stable and increasing population growth rates. But this difference was not statistically significant. So that was surprising too. When comparing seed dry mass, and for seed, the prediction was that a greater size of seed will offer the species a greater competitive ability because they have more resources, so they can grow faster or germinate or establish faster. That was the idea. I found that low abundance species with decreasing population growth rates had seeds that were on average more than 3 times heavier than species with stable and increasing population growth rates. But this difference was not statistically significant. So that, again, it kind of went against of my predictions. So that was seed dry mass. We're going to, I also wanted to know about seed bank duration because sometimes plants use that as a strategy, or some species might remain on the landscape stably because of seed bank duration. I found that low abundant species with decreasing population growth rates had 8 months shorter duration seed bank than species with a stable or increasing population rate. But this difference was, again, not statistically significant. However, there was a marginally non-significant interaction between the abundance categories and the population growth rate. Now let me report the results of the comparisons of common and low abundance species. So the next one won't have that break between decreasing and increasing population growth rate because I didn't have enough data to run those analyses. So I only compare common and low abundance. I compare leaf nitrogen because greater nitrogen content is associated to a strong competitive ability. There were no significant difference in leaf nitrogen content per leaf dry mass for common and low abundance species. I expected to find greater leaf thickness in common species because I hypothesized that they experience more herbivory damage than low abundance species. I found that the average of leaf thickness was found in low abundance species more than 10 times greater than common species. So the takeaway is there is not enough evidence to determine if there were any traits that could be used to distinguish a species with greater conservation need based on the cooperations of those 5 traits and the population growth rates. The big lesson I learned for this work was that we should increase the collection of trait data for the species that we have demographic information so we can do other analysis. I still think that we might find something if we have all the information. That could help us close some gaps in our knowledge. But for now, I was just focusing on saving one species at a time. Another thing was to conserve all the landscape. So when the first part of my dissertation was focusing on more like each species, what are the difference, but I know that in the last 10 years, the conservation literature has been thinking about like areas to conserve, right, to be faster. This is important because human populations continue to grow. For example, in Florida, it has been projected that by 2060, urban expansion will consume 2.7 million acres from agriculture and native habitat lands. One way to prioritize areas for conservation is to look at diversity measures in general. Phylogenetic diversity is a biodiversity measure that takes into account the evolutionary history of a set of species. Generally, it's calculated as the sum of all the branches. And for example, in this example, the bottom community has the lower species richness, but it has high phylogenetic diversity. This is kind of like you need to sum all the distance of the branches. That's what phylogenetic diversity is. Greater phylogenetic diversity has been associated to greater functional diversity because we expect to have a greater difference in traits in a set of species that represent different parts of the tree of life. There will be a difference in how fast they grow or how they take up nutrients, and that's why they think that we should prioritize greater phylogenetic diversity. So I wanted to quantify how anthropogenic activities decrease native biodiversity to a smaller set of species. For that, I was grateful to Dr. Kevin Robertson and Cinnamon Dixon that shared their data with me. They use a chronosequence approach. They identify areas that have the same fire return interval and fire season, and they only differ on the time after agriculture stops. So we have a reference native community, and we have different sites that have been used fire as a mode of restoration. And they also selected areas with other land uses, and I will include that at the end of my talk because it was such an interesting data set. So all of this is in the Red Hills. We're still on the same ecosystem where I did my experiments. I will also evaluate if after agriculture activities, like how fast we recover phylogenetic diversity and if we lose a specific set of species, meaning if those species represent a specific part of the tree or they are like over dispersed. And for that, I will use phylogenetic structure measures. Here, first, I'm going to show you the results of the species richness and phylogenetic diversity. Here we can observe that over time, there is an increase in the species richness and phylogenetic diversity. I just wanted to point out that the native community, I put it at the end of the graphs as a reference. Cool, so you can see that even after 100 years of restoration, we don't get to the same specific species richness or phylogenetic diversity. As I mentioned before, one of my objectives was trying to determine if we lose a specific set of species that are clustered, like a specific part of the phylogenetic tree, or is kind of like a random loss. So for that, I needed to use phylogenetic community structure measures. So there are different ways to determine structure. Phylogenetic structure, I use the mean nearest neighbor distance, which is the distance among tips. And I also use the mean pairwise distance, which is the sum of all pairwise distance between the species in a community. So here are my results of the first distance between the tips. The greatest distance was observed at sites where agriculture recently stopped, but it was managed with fire up to 5 years. So surprisingly, it seems like we recover this measure of phylogenetic structure really quickly. Within 5 years of management, we are practically recovered. Obviously, the native community has a lower distance. That means that you have a lot of the species that might be closely related to each other. And this is my mean pairwise taxon distance was the lowest on that recently managed sites. But if you look at the graph around 5 years or 15 years, we have actually just 5 years. You can see that it's almost the same phylogenetic structure than the native communities. And that was very surprising for me. Maybe it gave me hope that fire restoration was a good way to recover some of that diversity of the life in that area. So for my other question, because I wanted to know if we could lose this specific set of species, what I did is I compared the native community with the community that was managed for 100 years. And with that subset of species, I ran this model and I did find a significant difference, meaning that the species that we lost are closely related. So there is hope to find some information that will allow us to distinguish which species are the ones that we lost, and if these lost, maybe we can focus our restoration efforts in growing those species that even after 100 years of fire, we don't recover naturally. So as I said before, there was this very cool, and maybe I'm over time or I'm good on time. I think I'm good. Okay. So Dr. Kevin Robertson and Cinnamon Dixon also look at diversity of pasture and managed forests and pine plantation. So what I'm going to show you is the results of the phylogenetic diversity in those sites. And all of them are in the same kind of ecosystem, the red hills. So we have our native community, which is the never plow. 75 to 100 years of restoration is here, which is like the second better. To me, the most surprising part was the pine plantation had higher phylogenetic diversity than unmanaged forests, which makes sense because that unmanaged means like they have excluded fire. It hasn't been burned in the same frequency. Again, realizing that pine plantation was doing okay, I was surprised. So my takeaway number 3 is that plant communities do not recover to the same amount of evolutionary history even after 100 years of fire management, but they recover their phylogenetic structure after only 5 years, which was very surprising. A combination of fire exclusion and past history of soil disturbance had the greatest impact of phylogenetic diversity in these communities. Native communities had species more closely related to each other than expected. And I expect fire to be one of the principal filters driving this community, which is kind of obvious. But anyway, so this is the end of my talk. I think that this approach of thinking about specific kind of rarity and trying to connect all the dots has potential. Maybe we need better approaches, another set of species. Surprisingly, I think the lesson for me was that we should collect more trade data. It's not that hard, it's tedious, but not that hard. So we can make other kind of inference from this data. Analyzing phylogenetic structure was useful for my learning and also to maybe provide some encouragement for potential restoration efforts in the South. So yeah, that's it. Thank you. I'm done. And this is my acknowledgement for everybody that helped me in the field because that was a lot of work. Yeah, and that was my advisor on my committee. I studied at Florida State University. I'm done, so I'm going to stop sharing.